Hello, everyone, and welcome to the Osher Lifelong Learning Institute at UC Berkeley. Today is Ollie's speaker series, and we have a most remarkable colleague who is joining us, Berkeley Professor of Global Change in Biology, Bree Rosenblum. I'm Susan Hoffman, the Assistant Dean of Lifelong Learning at UC Berkeley. And speaker today, Bree, is the distinguished chair of Koshland for the Koshland chair, teaching and research. Sorry, I botched that so badly, Bree.
Great.
We're delighted that she's going to talk to us about from constraint to creativity, understanding resilience on our planet and in our lives. Bree has worked with Ollie in the past. We had the delightful time to work when she was working with undergraduate education and looking at ways to increase experiential learning with undergraduates, and has a very good sense of Ollie and also of intergenerational learning. Her work has been featured in the New York Times, the National Geographic, Discovery Channel, BBC, and on TEDx. She weaves her work on biodiversity of our planet with her passion, as you will see, with supporting others through her books, workshops, and her nonprofit called Hearth Institute. And so, Bree, thank you for joining us today. Thank you all. Please know that Bree will talk and there will be time for your questions. Post those questions in the chat. And as always, this is being recorded. We will caption it and post it to YouTube. So if you would like to share it with friends and others, you'll have a chance to do that. So Bree, the floor is all yours.
Thank you so much, Susan. And thanks everyone for being here. It's really nice to have the time with you. Ollie is one of my favorite communities at Berkeley, and I've really enjoyed time collaborating with Susan and working, I think, probably with some of you in our intergenerational mentoring program that we have. And today we are going to talk about some bigger picture things. I'm just going to share my screen and we will get started. So what I wanted to share with you today is really a bunch of different thoughts from my research and teaching about resilience. And I'm hoping that this is timely for everyone in terms of thinking about what's happening in our world in many dimensions. As Susan mentioned, I'm a professor of global change biology here at Berkeley. And what that really means is that I'm fascinated by our planet. And I am really fascinated by the question of how does life respond to rapid environmental change? And we will take a couple different angles on this together today. I'd like to just introduce myself to you first. I'd like to give you a vignette from my research on endangered species. And then I'd like to broaden out and share a little bit about why I think this work matters for you and your life, us and our lives. And then really just end on some big picture thoughts. So I'll just start with a brief introduction to myself. I have a long history at Berkeley. I started at Berkeley in the year 2000 as a PhD student, and my early work was at the White Sands in New Mexico. I'm curious, and I know that I can't monitor the chat well and also look at my slides, but if anyone would love to pop anything in the chat while we are going, that's great. I'm always curious if people have ever been to White Sands. It's, not immediately local, but it's a pretty fabulous national park in southern New Mexico. And I got interested in this landscape as an early graduate student because I was looking for places. I had just come back from living in Africa for a few years, and I was looking for places that I could drive to from Berkeley that had really dramatic environmental gradients that I could study how living things respond to environmental change. And the White Sands caught my attention because this is just an aerial picture. It's a relatively small formation, and it's surrounded on all sides by the Chihuahuan Desert. And the white sand dunes are geologically really young. They're just a couple thousand years old. And in that time, they have experienced a huge amount of change, and all of the organisms that live there have changed with the environment. And so I won't take a deep dive into any of these particular research topics. I just wanted to give you kind of an overview of what a global change biologist does, so that we can work up to talking a little bit more about our contemporary world. And so what I loved about White Sands is that there were all of these dramatic and very rapid adaptations. So I'm a huge fan of all the creepy crawly things on our planet. I love lizards, I love frogs, I love spiders, I love snakes. And so these lizards caught my eye because very rapidly, just in a few thousand years, they adapted to have color that matches the white sand dunes. And so their ancestors that are dark in color that live throughout the desert are nearby, but they have this special adaptation to avoid being eaten on the white sand dunes. And they're very well camouflaged. And it's not just the lizards. There's white mice and white crickets and white spiders and white kit foxes and white butterflies. And what I specialized in during those early years of my PhD was trying to understand from all the way down to the DNA molecules and all the way up to the ecosystem processes, how do living things respond to rapid environmental change? And I really loved getting under the hood and identifying the specific mechanisms that help animals adapt rapidly to environmental change. And at that time, it was really a brand new field to be using DNA and molecular techniques to study rapid adaptation in nature. And now we think of genes and genomes as second nature, but I just want to remind you that at that time, it was a really new thing to be doing because the first human genome wasn't even published until I was in graduate school. And that first human genome cost more than $3 billion to sequence and took more than 10 years to complete. So the fact that then that opened an era where we could use those same kind of techniques, not only to study humans and human health, but also to study all the other millions of species on our planet. So that was kind of my graduate school time. And what was happening right as I was finishing graduate school is that one of the colleagues in my lab at Berkeley, whose name is Vance Friedenberg, he's now a professor at San Francisco State, he was studying these mountain yellow-legged frogs up in the Sierra Nevada mountains. I'm also curious if anyone wants to pop in the chat if we have any avid Sierras folks in the audience. If so, you may or may not have seen these frogs. They used to be the most abundant vertebrate in all of the Sierra Nevada mountains. But over the course of the last century, they've been in decline for a bunch of different reasons that I'll share with you. At that time, right as I was finishing graduate school, these frogs had already been in decline, and Vance had been studying those declines that were due to things like introduced fish and other environmental changes. But right at that moment, something unusual was happening, and that was this. Vance was going up to his field sites and he was finding dead frogs everywhere. And this is extremely unusual if you've spent time hiking in the mountains. You don't see dead and dying animals. And at the time, it was at first unknown why this was happening. And then the culprit of these die-offs was identified. I'll share more about that in a minute. But what happened in my life trajectory is that all of a sudden I went from thinking about how life responds to rapid change on a geological scale, thinking about how animals can adapt to new environments over the course of hundreds or thousands of years. And all of a sudden I was thinking about, well, can animals respond to these kinds of environmental changes over years and decades rather than centuries and millennia? And I got really interested in whether I could use some of the genetic and genomic tools that I had developed and start applying them more to endangered species questions. And so when I got my first faculty position, which was in 2008, and I was at the University of Idaho for a few years, my lab focused on these two sides of what I would consider this ecological or evolutionary coin. On the one hand, I was still studying lizards and understanding how forms of life can adapt and even undergo the process of speciation when they're confronted with new environments. And on the other side, I was studying the declines and extinctions of endangered frogs around the world. And so when I got hired back at Berkeley in 2012, I was hired as a global change biologist, which really wasn't a field that existed at that time. It was kind of a People had been obviously studying climate change for a long time, but we didn't really have a framework for integrating our thinking about all the different environmental changes on our planet holistically and how those affect all the different species on our planet. So I came back to Berkeley with this mandate to have a bigger picture understanding in my teaching and research program to understand not only how life responds to rapid environmental change, but specifically what makes living systems vulnerable or resilient when the environment changes. And this is what I want to talk about today. What makes living systems vulnerable or resilient when the situation changes? Now, one of the other life-changing things that happened to me in this period was that right after coming back to Berkeley, I was hit in the head, literally. I had a massive concussion, and it took me months and months and months to recover from that. And in that period, one of the interesting things that happened in my professional life is that questions that had seemed very specific all of a sudden got very general. So I already had a long history of meditation and inquiry and a long love of nature. So I was very familiar and very comfortable with ideas about connection and the interconnectedness of all of our lives. But something about that head injury kind of rattled the cage a little bit and made me realize that the questions that I was studying about the ecology of our planet also had an incredible amount to say about questions of what makes us as living systems vulnerable or resilient at a personal level, at a collective level, again, at an ecological level, because I was already studying that, but also at a social level, educational, economic, and political levels. We are constantly experiencing change in our environment writ large. And a lot of the insights that I was gaining from studying endangered species felt like they could also be applied to our lives. And so over the last 10 years, I've done a lot of work not only on the biology side, which I'll share with you in a minute, but also in thinking about what makes our systems resilient. I've done a lot of work, as Susan mentioned, with higher education innovation. Some of that has been directly with Ollie. I do a lot of work with outreach with K to 12. I also do a lot of public outreach. As Susan mentioned, this is a movie that we did with the BBC a couple of years ago. And I also write books, textbooks in my field, but also I have a couple of popular press books, including a new one that just came out this last month, which I'm excited to share with you a little bit more as well today. So that's a little introduction to me and who I am and what I do at Berkeley. And what I'd like to do with the rest of our time together today is spend a little bit of time sharing my research on endangered species, and then spend a little more time talking about why I think it matters, even if you don't care about frogs, and even if you feel like we're confronting way bigger problems on our planet right now than whether frogs are doing well. So I hope to convince you not only that the frogs are interesting, but also that there's a reason why we should care about the lessons we learn from biodiversity, because we can apply them to our own lives. So let's come back to that moment in 2005 where my colleague Vance started noticing a die-off of frogs in the Sierra Nevada mountains. And I've already told you that I study both speciation and extinction. This is just a little cartoon for those of you that might not be as biologically minded about how we think about speciation on this planet. So we all share a common ancestor. Every living, breathing thing on this planet goes back to a single common ancestor about 3.5 billion years ago on our planet. And since that time, when life first emerged on planet Earth, we've had this incredible blossoming across the tree of life. And at the same time, throughout the history of our planet, we've had a process that prunes the tree of life. So speciation populates the tree of life, and then extinction prunes the tree of life. And this is an absolutely normal, natural process that's been happening for billions of years on our planet. And you may have heard the statistic before that 95% of all species that have ever existed on planet Earth have gone extinct. And that's true because species have a lifespan. They come to be and they go away. So all living things, including species, have a lifespan. And so extinction is a normal, natural part of our planet. That said, we are currently in a phase of extinction that is not normal. it is not exactly the way extinction has worked in the past. And that is because the magnitude and the rate of extinctions that are happening right now on our planet are far higher than they are in a normal period of life on Earth, even given the fact that there's massive fluctuations over time. We are clearly in an elevated period of extinction. And that's because of the rate of extinction, the magnitude of extinction, the pervasiveness across the tree of life, and most importantly, the cause of extinction. We have never, ever, ever before on planet Earth had an extinction that was caused by a single species, an extinction spasm caused by a single species. And so that's us. We as humans are causing an extinction spasm across planet Earth. And it is so large that it is already considered by scientists to be one of the big extinction crises on our planet's history. So if you've heard a little bit about extinction in the past, you might know that our planet has had 5 major extinction spasms in the past, including the KT boundary when we lost the dinosaurs. But this is the first one that is happening in modern times caused by our species. And so this idea of the sixth mass extinction was first advanced in 2011 and is now very commonly recognized and agreed upon by scientists around the world. And so I wanted to zoom in today. We could talk about extinction across almost any segment of the tree of life, but I'm going to zoom in today on amphibians. And I am going to promise you that there is a hopeful note at the end of this. And so amphibians are an incredibly diverse group of vertebrates around the world. And they are currently experiencing extinction rates that are more than 100 times higher than we would expect them to be having if humans weren't changing the quality of our planet. And as you might know, that obviously there's a lot of different factors that are contributing to global change right now. Obviously, we have loss of habitat, which impacts species around the world. We have contaminants and pesticides that are polluting waterways, which is particularly important for organisms like amphibians that breathe through their skin. We have climate change, which has a pervasive effect all the way from the depths of the ocean to the upper reaches of the atmosphere. We have introduced species that either compete with or eat native species. And then we have a wide variety of pathogens and pests that are moving around the world a lot faster than they ever have before because of our human transportation networks. And so what I want to do today is zoom in on this question of emerging infectious diseases. Obviously, all of these other stressors are important for amphibians, but it turns out that the culprit of those frog die-offs that I had mentioned is this particular fungal pathogen called the chytrid fungus. And this is just a little video I took under the microscope. The chytrid fungus is an aquatic fungus. These little single cells are called zoospores. You do not need to remember that, but they're basically like little cells with a flagella. They swim around in the water. And when they find a frog, they burrow into its skin. And then they mature into this other life stage, these big round balls. You can see that they're reproducing more zoospores inside. And that process of the fungus attacking and embedding itself into the frog's skin can kill frogs. And this is a pattern that we see in endangered frogs all around the world. It's a simple time process where there's no chytrid in the environment, and then chytrid arrives, and shortly thereafter, frogs decline, disappear, or go extinct. So not all species are equally susceptible, and not all environments are equally friendly to the fungus, but we are seeing the chytrid's effects all around the world in hundreds and hundreds of species of amphibians. And I'll just take a closer dive into the Sierra Nevada mountains for a minute to give you a sense of how quickly this pathogen is killing frogs. So this is just a picture map of a single lake basin in the Sierras. It's called 60 Lake Basin. You might guess that there are about 60 lakes. And this was 2004, the year I was working to finish my PhD. And there were frogs present throughout the basin. And most of those lakes were green, which means that there were frogs present, but no chytrid there. And the scientific name of the chytrid fungus is Batrachochytrium dendrobatidis. You don't need to remember that, but that's why my slides sometimes say BD, is because we abbreviate the scientific name that way. So that's how the situation looked that first year. The year I finished my PhD, we started having chytrid present in these yellow lakes, and it was spreading fast. And every year we could track the spread of the chytrid and the death of the frogs in the wake of that spread. So the next year we had more frogs going extinct. Those are the black lakes, and the gray lakes are the ones that are starting to be highly impacted. And then in 2007, we basically had almost no places where we had healthy frogs left. And so that's where I came in. And I wanted to just make the point that this is not just something that's happening in the Sierras. This is also a photo that I took in one of our field sites in Panama just before the pandemic arrived. We were there surveying for frogs, and these are forests that used to be absolutely vibrant with frog songs, and they were quiet. You could hear a pin drop at night. And it's not just that we have large-scale disappearances of amphibians around the world. It's also that we have specific losses. So this is a specific frog that I was holding while we were in Panama. It is the last frog of a species that had never been described to science. So my colleagues had found this one frog in the forest, recognized her as not being a frog that they had ever seen before, and they had no idea what the species was. So they brought her back to the lab so that they could describe the species and her species could be known to science. But they couldn't figure out exactly what she was because they kept going back to the forest year after year after year and not being able to find any others of her kind. So by the time I got there in 2019, this frog had been in captivity for years. She was dying, and she was literally representing the death of a lineage of our plant on our planet that we didn't even know what was. An undescribed species was being lost. And these are the kinds of things that are happening all around the world, not only with frogs, but with other species. And so when I got involved in the frog research, I was really interested, again, in this overarching question we're talking about today, which is what makes living systems vulnerable or resilient to rapid change. And if we rephrase that for the frogs, the specific research question here was what makes frogs vulnerable or resistant to these kinds of disease-induced extinctions? And so what I did, which seemed logical at the time, was I started by studying the most vulnerable frogs I could find, the ones that seemed to be disappearing the fastest, because they seemed like the ones that were of greatest conservation concern. So I took a similar approach to that which I had done at my White Sands research, which is to use genetic and genomic information to try to recreate the history of the spread of this fungus, to try to understand specifically what makes some frogs susceptible to these diseases, and to understand whether there were specific targets for conservation that we could apply by understanding resistance and tolerance to this disease. And so rather than show you a bunch of data, I'm just going to tell you the conclusions from this phase of the research. We used genetic data to reconstruct the history of the chytrid fungus around the planet. It's very similar to what epidemiologists do when we reconstruct COVID-19 trajectories and spread, or we reconstruct different strains of the flu and how they move around the world. So we use those kinds of molecular epidemiology approaches to reconstruct the history of chytrid around the world. We found not only that chytrid is on all continents with amphibians, but we also found that not all chytrid strains are created equally. There is a specific chytrid strain that's the most deadly and the one that is spread around the world the most quickly. And yes, it looks like humans do have a role in that spread, but once that chytrid starts spreading, it really takes off and it can get into very remote places very quickly. So on the frog side of the story, what we learned is that the frog immune system does a very bad job detecting that it's even infected. The chytrid fungus is kind of hiding from the immune system. And what that means is that frogs basically are getting sick before they can mount an appropriate immune response. And when we compared frogs that were dying from chytrid all around the world, we found that the frogs that were the most vulnerable had very specific things in common. They had specific immunological traits, specific physiological traits, specific life history traits. We could start to predict which frogs would be the most vulnerable to chytrid. So scientists love predicting things, but we love it even better if our predictions can help us create actions that are going to protect endangered species. And the problem with this line of research is that it didn't really help us, because even though we could identify the specific reasons these frogs were getting sick, there was no way to scale our interventions. All of the ways that we knew about to protect frogs from the chytrid fungus and to cure them once they were sick were incredibly time intensive, incredibly cost intensive, and very, very difficult to scale. You cannot take all the 7,000 species of amphibians into the lab, clear them with antifungal drugs, and then put them back out into their environments. And you also can't fly armies of undergraduates around the world and have them hike to high alpine lakes and administer individual treatments to individual frogs. So it just was not scalable. And this was a very depressing time in our research. The interesting thing is that right at this point, when things seemed so depressing, I had a little epiphany, which is that I'd been studying the wrong half of the equation. I had been spending all my research time studying the most vulnerable frogs instead of studying the frogs that were the most resilient. And we had a massive change in the focus of our research program and started thinking about, wait a minute, instead of looking at what makes vulnerable species vulnerable, why don't we look at what makes resilient species resilient? And so I just want to tell you a very hopeful anecdote on the resilient species side. This is my collaborator, Roland Knapp. He is an amazing scientist who has been hiking in the Sierras for his entire adult life. And in 2012, he had an idea, which was that all of the frogs in this part of the Sierras had been lost long ago. But there were very closely related frogs, the same species, in nearby ponds. And so he, on a whim, a well-planned whim with appropriate permitting, decided that he was going to hike 20 frogs from one lake to another. And this is the maximum number of frogs that he could fit in his bear box in his backpack that season. And so he brought 20 frogs from one pond and put them in another. And he left them there and didn't do anything, didn't treat them with any antifungal drugs, didn't do any other manipulations in the environment. He just left them there. And he did that in 12 lakes, so it was a replicated experiment. And so I already told you that the trajectory that we were seeing over and over again in the Sierras and around the world was that when chytrid first arrives, frogs decline. Well, now what we were seeing is that once the frogs had declined, and Roland hiked the frogs in, so this was when he did that right here in 2012, the frogs absolutely took off. And in the course of just a few years after they had gone through a massive decline, they rebounded to incredible levels, hundreds and hundreds and hundreds of frogs per lake. And you'll see that then it tapers off just a little bit, but these populations are stable. And so I just wanted to show you, this was just a couple months ago, Roland and I hiked up to this same pond that I showed you at the beginning. And this is him crawling very quietly so as not to disturb the frogs on the bank. And if you zoom in, once we got to the very edge, we were hiking over to this little sandy bar, and we got to the edge and we peeked over the edge. And what we saw there was this little beach full of frogs. Hundreds and hundreds and hundreds of adult frogs and thousands and thousands and thousands of healthy tadpoles. And so all of this is from a single isolated reintroduction event. And reintroductions are a valid conservation strategy. This just wasn't just like an idea Roland had, but this was an incredibly low cost reintroduction. It was Roland carrying 20 frogs in a backpack. And so what I wanted to highlight here is that it's not only that the frogs started coming back, but once the frogs came back, all of a sudden, and I don't know if anyone notices what I'm about to point out in this picture, but all of a sudden, other vertebrates started to come back. Snakes started to come back, coyotes started to come back, bears started to come back, because frogs are a pivotal member of the ecosystem. And it's not only that this happened in this one lake. Out of the 12 lakes that Roland seeded with a small number of frogs, 9 of those lakes have shown these kinds of massive recoveries. And so one of the things that we're now doing is we have this big grant across a bunch of institutions, which is cheesily called RIBITR, but that stands for Resilience Institute Bridging Biological Training and Research. And what we're now doing is, instead of comparing all of the most vulnerable species around the world, which we're still doing, but we're now focused on all of the most resilient amphibian species around the world to try to understand what are the similarities among those species. And so our Sierra Nevada site is being compared to sites on the East Coast. We have sites in Panama and sites in Brazil. And so what we're seeing in those communities, and again, I'll just summarize some of the research here instead of showing you the actual data, is that frogs can recover following these chytrid-related declines. And I showed you that in photographs from the Sierras, but here's more of a data slide from our field site in Panama, the one that I showed you that had a quiet forest. And before chytrid arrived, this is just a bunch of different frog species. So I'm showing you the community level. We had all of these species in kind of their historical high abundances. And then chytrid arrived, we call that an epizootic, the same way that we'd call it, epizootic or for human disease, same concept, right? We have this massive die-off of frogs. And then we have the recovery, and the chytrid is not gone. It's just that the frogs are now living with the chytrid fungus and doing fine. Now, why are the frogs living with the chytrid fungus and doing fine? Any guesses? The fungus isn't gone. The fungus is no less virulent. It's that the frogs have adapted, right? So the resilient frogs share very specific adaptations. And what my job has been to study the genomes of all of these frogs around the world and identify specific genes that have undergone adaptive evolution. And what we see is that the frogs are adapting, and I'm not going to bore you with details about the immune system, but we can actually pinpoint what parts of their immune systems have gone through these genetic changes. In MHC, the major histocompatibility complex, that's also a really important element in our own immune systems. They have changes in other genes like RIN3 and this GVINP1. These are genes that are known to be involved in tissue healing, known to be involved in disease resistance. And so we can actually track the ways that these frogs have adapted. Now, here's the extra good news about this, is that adaptation is something that the frogs are doing. So we are not flying undergraduates around the world to hold frogs in little plastic containers and administer antifungal treatments. The frogs are adapting, and this is exceedingly scalable because it's waiting for a natural response. And the important thing here is that we can then study those natural responses. So Roland hikes frogs in, the frogs go into a new lake, and if those frogs survive, they are surviving because they are adapting to the threats. They are changing genetically so that their offspring are going to be better suited to survive in those conditions in the future. And then if those offspring increase, now we have a way to study over the course of many years how this adaptation process works. And so conservation efforts now can shift away from trying to get rid of the fungus to actually buying time for frogs to adapt. And we can use this very simple, very inexpensive strategy of assisted migration or translocations, where we're moving frogs from healthy populations into populations that need to recover. And we can do this in a way that really doesn't cost a lot, not only in terms of funding, but also in terms of our own conservation ethics. Because you can imagine, if you compare this strategy with other strategies like these kinds of pods where people are trying to, for millions and millions and millions of dollars, trying to breed resistant frogs, but then those frogs are completely maladapted to the environment. And so we're spending all this money trying to breed frogs that then die when we release them into nature. We're not doing that. We're letting the frogs have their own adaptation process in their own natural environment. And if you also compare this to other kinds of interventions in the conservation arena where we're trying to manipulate the genetics of organisms, where we're even, and I'm saying we as a community, not me in the we, where we're even trying to bring back extinct species like the woolly mammoth. There are a lot of fascinating questions in how we should be using our conservation dollar, but I think that some of these questions are very easy to answer when the organisms can provide the very genetic changes that we would otherwise be trying to induce by spending a huge amount of manipulation and money. Okay, so let's take a break from frogs. I'll just tell you one big picture lesson from all of that research, and then I'll try to apply it a little bit more to our own lives. So what I've told you so far is that I've been studying this big picture question about what makes living systems vulnerable and resilient to rapid change. And I do this in lots of species around the world, even though I just shared a single little frog vignette with you that has a kind of happy ending in the sense that we were able to identify what makes some frogs resilient and how we can use that resilience to help recover species from the brink of extinction. But what I didn't tell you is that all of our research suggests that there's a handful of very general processes that lead to resilience. And there's really only three things that determine whether a species is going to be vulnerable or resilient to environmental change. And I want to make it clear that this applies not only to frogs, but it also applies to us. So we can ask this question, what makes living systems, including us, vulnerable or resilient to environmental change, but also vulnerable or resistant or resilient to personal, political, economic, and social change. We are all part of a living system. Even our social systems were created by us, living beings. And so our systems evolve all the time. We evolve, our systems evolve. And the things that define whether we are going to be resilient or vulnerable are the following. So let's take vulnerability and resilience as the thing that we want to predict. There's really three ways we can predict whether a living system is going to be vulnerable or resilient. The first is what's their exposure to environmental change. So the magnitude of the change affects how we're going to respond. That's obvious. It's obvious for species. If you hit them with 1 degree of climate change versus 5 degrees of climate change, that's a big difference in exposure. The same is true in our lives. If we have a relatively modest amount of change, or an enormous amount of change in the same period of time, that exposure risk is totally different because we can adapt and adjust to moderate changes much better than we can adapt or adjust to radical changes all at once. So we can address exposure risk, and we should. We can also address sensitivity. So our sensitivity to environmental change is something that is essentially innate, right? Some species are just going to be more or less sensitive to changes in temperature, for example, than others. Some species are going to be more or less sensitive to a particular disease. The same way that all of us have particular areas of sensitivity and particular areas of robustness. I'm sure every one of you on this call can think of at least one thing that you're particularly sensitive to, and one thing that you have a little bit more robustness to handle. And so your exposure risk and your sensitivity are really key factors to whether or not we species are going to be able to adapt to change. But the single biggest thing that I think is often overlooked in the conservation literature, and I think is important in our own lives, is response. So you or a frog can be exposed to the same risk. You can have an innate sensitivity, but you can still have a response. And that response in our frogs was the ability to adapt to the chytrid fungus, have actual genetic changes that persist across generations that make certain frogs' progeny more suited to survive with a continued threat. And that process obviously is happening for humans as well. We're still evolving and adapting. But when I think about it for humans, I mean it in a little bit more of I mean it less in a biological adaptation way that we have to wait 3 generations till our children can handle something better. I mean it more in terms of adaptation to the conditions of our lives, is that we can change, we can respond. And that if you look at the last 25 years of my own research, when I look at how much exposure matters, how much sensitivity matters, and how much response matters, response is the thing that matters. Response is the thing that can save a species from the brink of extinction. And response is the thing that can change the way we relate to our lives and our own social systems and our own stressors and our own environments. And so what I would say after 25 years of studying biodiversity, both on the adaptation and speciation side and on the declines and extinction side, what I believe in, what I have faith in, what I think is absolutely upheld by all of the data is that we have a phenomenal amount of evidence for life's adaptive potential. That is not a Pollyanna statement. That is not a wishful thinking statement. That is a fact about life on this planet. And it is a fact about ourselves as well, not just other species, because we are a species on this planet. And because I have such incredible faith in life's adaptive potential, I have very little patience for these kind of like boring linear narratives that make it seem like humans are just the be all end all. And I also have very little patience for narratives that make it seem like humans are the absolute villain in the story on our planet. And I'd like to just give you a little bit of a perspective about why I think understanding response and evolution matters for contextualizing where our own species is at in the history of our planet. So for those of you that don't know a lot about human evolution, and I know this is a little bit of a jump from frogs to humans, but I'm going to tie it all back together for you in a minute. For those of you that don't know as much about human evolution, there have been more than 20 species of humans that have walked this planet. Sometimes those species have actually overlapped in space and time. And sometimes they have been sequential in different parts of the world at different times of our history. Our species, Homo sapiens, is less than 500,000 years old. And the reason why this is important to me is because I told you at the beginning, when I showed you that tree of life, that all species on our planet have an expiration date. All species have a lifespan. And the average lifespan of most species on our planet is between 1 and 10 million years. So if we take that, it's not a promise, but if we take that as a general average across the tree of life, our species is a young species. And I like to think of our species, Homo sapiens, as an adolescent species. We're 500,000 years in. Who knows if we're going to have 1,000,000 years or 10 million years, or we don't know, but we are a relatively young species. And I have adolescent children. This was a picture of my son's room. In a time I should have been way more on top of him, but wasn't. And when I went up one day and was like, oh my goodness, you need to get yourself under control. I was like, this is exactly how I feel about our species, is that I think of us as an adolescent species, and we are making adolescent decisions in some arenas of our relationship with ourselves, each other, and the planet. And I love The Matrix. I'm not sure if any of you have seen it. We're getting close to the end here. I know that I want to leave a few minutes for questions, but I think we have a really important choice as a species right now, which involves our choice to grow up. And that means that we have to reckon not only with our world, but we also have to reckon with our worldview, because I am a firm believer that how we see things and how we think about things is what creates the reality of our planet. And we are living in a very fractured world that is coming from a very fractured worldview. And to me, the antidote to that is considering the possibility that our worldview needs to be reoriented towards one of connection. Now, you're going to initially probably feel like that sounds like a very knee-jerk positive thing to say at a time when we can't just be knee-jerkly positive. But I will stand behind this, and I will tell you why. And I don't want it to seem like I'm neglecting some of the important facts. because there will be some species that can't adapt to changes in our planet in time. And there will be some of our own social, political, and educational systems that will fail. And so when I say we have to reckon with our world and our worldview, I don't mean that this is going to be easy, but I do think that reckoning with connection is a much more interesting thing to reckon with than reckoning with a fractured society and a fractured worldview. Because for me, the very fundamentals of our life are built on connection. And I think this is utterly indisputable. As a biologist, it is indisputable. And so that's why I want to zoom out for our last few minutes and tell you why I think it is indisputable that we are built on connection, that we have a phenomenal amount of resilience, and that all of that hinges on our responses to changing conditions. So we are built on connection in the very fabric of our DNA. Our DNA is held together literally by bonds, trillions and trillions of bonds between molecules. And our DNA in our cells is inside of a nucleus, which formed billions of years ago by one life form learning to live inside of another life form. So our cells themselves are the result of a symbiosis that goes back billions of years. And all of our cells that comprise our own bodies are an incredible complex orchestra that works together despite its differences. And again, I don't mean that in a knee-jerk way. I mean that in a profound biological fact way. In fact, you yourself are an ecosystem. We know that your body is less than 50% made up of human cells. That is a fact. Your body is comprised of bacteria, viruses, fungi, and of course, human cells. But you yourself are an ecosystem. You are built on connection. You would not survive if it was not for all of the connections that are made in your DNA, in your cells, with other species living inside you. And the same is true at a larger scale, at the ecology of our planet. You cannot have a forest without what's happening not only with the trees themselves, but with all of the life under the soil, with all of the dispersers and pollinators in the sky and in the forest canopy. And it's not just the ecology, it's the evolution. I told you in the beginning that we are all descendant from the same common ancestor. That is not just true of our own species, it's true of all species on our planet. Evolution is the result of interconnection, and we are at the leading edge of 4.5 billion years of history on our planet. And so for me, that leading edge, that we are literally the front edge of an expanding universe, is a point of inspiration. It's not a point to think, oh, well, we're at the beginning of the end and everything's terrible. And I know that we're all facing lots of challenges personally, planetarily, socially. But if we maintain a perspective that what's true and what matters is the fundamental reality of connection, I think that that gives us a real potential to think about what are the things that we value more than fear. And I think for me, I'm using the word love instead of connection, just to be a little bit more, to make a slightly more aggressive point on this, is that I think we have to decide what we love more than what we fear. Because if we don't make those decisions, then our species doesn't grow up. And we end up in the vulnerable category instead of the resilient category. And so I'll just close before we open up for a little bit of conversation and question with a few things about what's my leading edge and a few invitations for you. So I have a couple new books that just came out this month where I go a little bit deeper into this philosophy about connection and where I weave together stories about science and personal growth to think about how do we really make these transitions from constraint to creativity. So I invite you to join me on that book journey. I also wanted to mention that as part of the expansion of my own work in higher education and intergenerational learning, that I have a nonprofit, an educational nonprofit that started, and we are actively launching and building facilities for intergenerational field courses. And I'll just give you a quick taste of that. We have 80 acres on the edge of the Rio Grande in in northern New Mexico, outside of Taos. And we have this amazing off-grid kind of hub where everything is centered out of. We have this phenomenal university caliber telescope so that we can give people experiences from the Rio Grande River through the high desert land up to the sky. And this is my first photo that I took from that telescope when I learned how to use it. This is a nebula inside the constellation Orion. And it, to me, offers a huge amount of inspiration as I'm thinking about what does it mean to be on the leading edge of our lives and of our times and of this time in the history of the universe. For me, it's really fascinating to be able to literally look up into the cosmos and muse on some of those questions. And so I'm curious for all of you. This is a little bit of what's at my leading edge in terms of my interest in the biodiversity of our planet and also in how we can apply the lessons from biodiversity science into our own lives. And I'm very curious to hear a little bit what's on the leading edge of your worlds. And so I would invite you to stay in touch if you'd like. My email address is here on the slides, and Susan can certainly share it. I have a mailing list on my website. I would love if you wanted to have a conversation about any of these topics. And I also would love if anyone is actually interested in either supporting or being part of the work we're doing at the Hearth. We have a couple of really fun, both online and in-person retreats coming up, and it would be delightful to stay in touch with the OLLI community about all of that. And so I will stop sharing and bring us back together and take a few minutes in conversation.
Bree, thank you very much. What an incredible presentation and what nice kind of invitations. We've got a few questions, and I want to, one that starts with Susan asking, can you please clarify, was the same species reintroduced to the lakes, or was it a non-impacted species that could thrive?
Yeah. Thank you for that clarifying question. That's great. Yes, it's the same species, just from nearby lakes, but the difference is that the individuals that reintroduced had already survived the chytrid wave. So those individuals that were reintroduced were essentially pre-adapted. They were already the survivors. So your question is fabulous. It's not that the frogs then adapted just in 4 years. It's that the frogs had adapted over the course, it had only been probably 10 years, but that's a lot in frog generation time. And so those frogs had adapted over the course of those years. And then the survivors that were now re-flourishing were moved. And so they were kind of pre-adapted to be able to survive even with that persistent threat. It was a fabulous question. Thanks, Susan.
That's great. And then Eliana has a comparative question. How did the diversity of frog species compare between the original lake and the lake with the adapted frogs?
I love these fantastic biology questions. Thanks for, it's so funny because when I share with an audience like this, I kind of feel like, well, nobody's actually going to care about the biology. But so it's lovely that some of you do. Thank you. So the Sierras are really interesting because it's not like in Panama where we might have, 20 different species of tropical frogs all coexisting. The species in the Sierras, there's really only a few species of amphibians and very few species of frogs. So this is the only species of amphibian in those high alpine lakes. They're the ones that get the highest. We don't get the lower elevation red-legged frogs, even the tree frogs. Most of the other amphibian species don't make it that high. And so that's partly why this one species is such a keystone species in those ecosystems, is because it's not like, oh, well, if this species declines, there's 10 other frog species that kind of serve a similar ecological function. And that's why the recovery of this species means so much for those high alpine environments.
Fantastic. And then Patricia has a question similar to the others, that how the frogs adapted to a different lake than their original ones. Was it the actual better frogs selected randomly? Does that make sense to you?
Yeah. So that's a great question, Patricia. And I think it's similar to what we were talking about with Susan's question at the very beginning, is that even though I didn't explain clearly enough that the frogs weren't selected randomly, they were, survivors from a prior epidemic that then were transferred, that they were pre-adapted to those conditions and so have been doing just phenomenally well.
And And then do you see Ileana's last question? Okay, go ahead and repeat
Yeah.
it.
Yeah, absolutely. So I would say that the connection here for me is partially literal and partially metaphoric. So when we talk about, so adaptation for me has two components. There's biological adaptation. When we, as evolutionary biologists, talk about adaptation, we're talking about genetic changes in a species that are passed across generations. So that is a very narrow definition of adaptation. That is what we mean when we talk about adaptation as scientists. For me, when we talk about adaptation as humans, we're talking about something a little bit more metaphorical. We're talking about the changes, the internal changes that we need to make that are going to create an environment where we can be better adapted to the conditions of our planet. So I'm not saying that, so the metaphorical part is not to say that, oh, we need to induce genetic mutations so that we can survive better when it's 5 degrees warmer. That's not what I'm talking about today. I'm taking a kind of metaphorical leap where the connection between the two halves of my presentation for me is really more of the framework about resilience, that when we're thinking about resilience in not only biological systems, but also human systems, that we need to be thinking about exposure to threats. We need to be thinking about our sensitivity to that exposure, and we need to be thinking about our response. And for me, the most important of those three, not just in the science, but in our lives, is our response. And for me, that's a positive, inspiring idea because that's actually the only one of those three that we have any control over. Especially at a time right now where I know a lot of people are feeling like anxiety is high. There's a lot of things that we don't have control about that really matter in our world. And that if we cannot limit our exposure to stress and we cannot limit our sensitivity to that exposure, then the thing that we can and need to work on is our response, both individually and collectively.
Yeah. Well, obviously, Bree, what I begin to think about as somebody with more of a political philosophy background is the way in which the disruptors have taken over the worldview and not given anybody any time, and who seem to have the assumption that, well, some species will die and that will be okay.
Yeah. Yeah. And I think that also maybe gets a little bit at, I know we're just coming to the end of our time, but to Jessica's comment in terms of what might some next steps look like, for me, I think that there is a phenomenal amount of deeply negative and constrained thinking that we are fed constantly in our society that we internalize. And I think that my focus on what does it look like to actually nurture and nourish and foster connection is not just a platitude. That's actually really hard work. And I think that map between what we're eating, metaphorically, like what we're taking in, we're internalizing from our culture, and where we're putting our attention is really, really, really important right now. And that's really why I actually had been writing a much different scholarly book last year. And that's why I scrapped it and wrote this kind of short, sweet book that's called And, because it's all about what do I think we need to do internally, each of us, to make that investment. So instead of just imbibing the negativity, we're actually making a conscious choice to spend more time nurturing connection and creativity in our lives.
Great. I think people are going to be very interested in the book. I think also it seems like the medical world has begun to shift slightly from the disease model to the prevention and looking at what does make people resilient. So we have the biological sciences partly to thank for that. So, well, if there any other closing remarks? You're getting a lot of kudos in the chat. So
well, I've just absolutely loved all the times I've had a chance to interact with the Ollie community, and so excited to have a open channel to chat with folks that were here today and just keep possibilities for future interaction and collaboration open. I always love working with you, Susan.
Yeah, and likewise. If I can hold people's attention just for one more minute, I want to let you know that we have 5 remarkable events happening over the next 3 weeks. Please take a look at your dashboards at the Ollie website, but let me just say very briefly that next Thursday we have a vital aging session with Annette Siegel to talk about the life-changing benefits of living abroad. So one adaptive strategy. The other is then on Friday, a week from today at the same time at 10:30, but also it will also be in person. We have Dorothy Lazard, who is Oakland's celebrated librarian, now retired, who has written a book, and she's going to come and talk with us about placing yourself in history, writing memoir. And so please take a look at that. Dan Camden is going to be with us on February 21st. That's titled Science and Policy of Energy and Climate Justice. As you know, he is our foremost energy scientist on campus who has now moved to the East Coast in Johns Hopkins, where Bloomberg has put $6 billion into a new research center. So come and hear Dan speak. You will also have a chance to hear Jeff Ruck, who has been a really well-established whistleblower, who is going to talk to us about the impunity of official disinformation. He's worked on many administrations, and he will have a perspective on what we are encountering right now and what we have in the past. So I hope you will join us for one of those events, and also join us on February 19th when we celebrate Stephanie Wells with an Excellence in Teaching and Learning Award, which will take place at Freight and Salvage at noon on February 19th. Thank you, everybody, especially thanks to Bree for a wonderful presentation today, and we'll see you next week. Take care.